Before there were hospitals, before microscopes, before anyone knew what a gene or an antigen was, there were patterns that people noticed but could not explain. Families where the first child lived and the second died. Villages where miscarriages seemed to follow certain women like a shadow. Blood transfusions that saved one life and ended another in minutes. For centuries, these events were folded into superstition, prayer, fate, or punishment. No one suspected that the explanation lay in something so small it could not be seen, a single missing marker on the surface of a red blood cell, silently shaping lives across generations. Tonight we begin a long journey into that invisible world. Right now, as you breathe and listen, blood is moving through you with quiet precision. Millions of red cells flow through your arteries, carrying oxygen, removing waste, sustaining thought itself. They all look the same under a casual glance, smooth and simple, but on their surface is a complex language made of proteins, sugars, and molecular signals. Most humans share a particular marker in that language, a sign that says self, recognized by immune systems across our species, but a minority of humans do not carry it. About 15% of the world's population lacks this marker entirely. They are Rh negative. At first glance, this seems unremarkable. Humans vary in countless ways: eye color, height, skin tone, metabolism. Why should blood be any different? But Rh-negative blood is not just another cosmetic variation. It is different in a way that matters deeply. It can turn pregnancy into a battlefield. It can determine who lives and who dies in an emergency room. And its distribution across the world follows a pattern so strange that it has fueled scientific debate, historical speculation, and myth-making for decades. Every primate species we have studied carries some version of the Rh factor. Chimpanzees have it, gorillas have it, orangutans have it. Monkeys, apes, even distant mammalian relatives carry related proteins that serve similar functions. Across the evolutionary tree, this marker is almost universal, and yet in humans alone, a significant minority simply does not have it, not damaged, not altered, missing. Evolution usually does not work this way. Traits that are deeply embedded across related species rarely vanish in only one branch, and when they do, they rarely persist at high frequency unless there is a compelling reason. That is the first crack in the story, the moment where curiosity turns into unease. Why would humans lose something so widespread, and why would that loss survive? The mystery deepens when we look at where Rh-negative blood appears. It is not evenly scattered across humanity. It clusters. In some populations, it is almost non-existent. In others, it is shockingly common. Among the Basque people of northern Spain and southern France, nearly 1 in 3 individuals is Rh-negative. In Ireland, Scotland, Wales, and parts of western France, the numbers are far higher than the global average, but move east into Asia and the trait almost disappears. In large regions of Africa, indigenous populations show Rh-negative frequencies near zero. Among indigenous peoples of the Americas before European contact, it was virtually absent. This is not random. This is not what you expect from a simple mutation drifting freely through the species. This is a geographic signature, a genetic footprint pointing towards something in the human past that shaped who carries this blood and who does not. For most of human history, no one knew this pattern existed. The story only begins to surface in the 20th century, when blood itself finally started to give up its secrets. Before that, doctors knew transfusions were dangerous, but not why. Some patients recovered, others convulsed, went into shock, and died. Blood seemed both miraculous and treacherous, a substance that could heal or kill depending on invisible rules. In the early 1900s, one scientist finally cracked part of the code. By identifying the ABO blood groups, he showed that blood was not universal, that the immune system could recognize foreign blood and attack it violently. This discovery saved countless lives, but it was only the first layer. Beneath the ABO system lay something even stranger, something that would not reveal itself until decades later. In 1940, researchers conducting routine immunology experiments noticed something odd. Antibodies produced against monkey blood reacted with most human blood samples, but not all. Roughly 15% of the samples showed no reaction, as if the red blood cells were invisible to the antibodies. The researchers had stumbled onto the Rh factor, named after the rhesus monkeys used in the experiments. If you had the marker, you were Rh positive. If you did not, you were Rh negative. At first, this seemed like a technical detail, another classification for lab work and transfusion safety. But almost immediately, doctors began to see connections. Cases of newborns with severe anemia, babies swollen with fluid, their skin yellowed by jaundice. Patterns that had baffled obstetricians for generations suddenly made sense. An Rh-negative mother carrying an Rh-positive baby could become sensitized to the baby's blood. During childbirth, tiny amounts of fetal blood could enter the mother's circulation. Her immune system, encountering the Rh protein for the first time, would identify it as foreign and create antibodies. The first child was usually safe, but the antibodies remained. And in the next pregnancy, if the baby was again Rh positive, those antibodies could cross the placenta and begin destroying the fetus's red blood cells from within. This condition, hemolytic disease of the newborn, was devastating. Before modern medicine, it killed countless infants. Some mothers lost child after child, never understanding why their bodies seemed to betray them. Evolution, which is supposed to favor traits that promote reproduction, appeared to have allowed a blood type that actively endangered it, and yet Rh-negative blood persisted. This is where the questions sharpen. If Rh-negative blood offers no advantage, why did natural selection not eliminate it? If it is harmful, why is it so common in certain populations? And if it is ancient, why does it not appear evenly across the world? As scientists mapped Rh-negative frequencies across continents, a picture emerged that was both clearer and more troubling. The highest concentrations appeared in regions that were geographically isolated for long periods of time: mountain valleys, Atlantic fringes. Places where ancient populations could survive waves of migration without being fully absorbed or replaced. The Basque region stood out above all others. Not only did the Basque people have unusually high Rh-negative frequencies, they also spoke a language unlike any other in Europe. Euskara does not belong to the Indo-European family. It has no known relatives. Linguists have tried for over a century to classify it, to link it to ancient Iberian tongues, to languages of the Caucasus, to North Africa. Nothing fits. It stands alone, a linguistic fossil preserved in the mountains, while every other pre-Indo-European language in Europe vanished. Genetically, the Basque also showed signs of deep ancestry. Certain markers suggested continuity with some of the earliest modern humans in Europe. While surrounding populations changed through migration and mixing, the Basque remained relatively isolated, preserving traits that elsewhere were diluted or lost. This combination of linguistic isolation, genetic uniqueness, and high Rh-negative frequency made the Basque a focal point of speculation. Were they survivors of an ancient population that once covered much of Europe? Did Rh-negative blood represent a genetic echo of Ice Age humans who found refuge in these mountains while glaciers advanced and retreated across the continent? Similar patterns appeared elsewhere. Celtic regions along the Atlantic coast showed elevated Rh-negative frequencies. Mountain populations in the Caucasus, Berber groups in North Africa. Again and again, Rh-negative blood appeared where isolation had preserved older genetic layers. One explanation seemed plausible. Rh-negative blood might be ancient, older than many modern populations, preserved in refuges while later migrations reshaped the rest of the world. The Indo-European expansions, the spread of agriculture, the rise and fall of empires could have diluted Rh-negative frequencies across much of Eurasia, leaving pockets where it remained strong. This explanation fits the data reasonably well, and yet it does not fully satisfy. Because even in small, isolated populations, traits that severely reduce reproductive success tend to fade. Genetic drift can amplify random features, but pregnancy loss on a large scale is a powerful counterforce. Something must have allowed Rh-negative blood to survive long enough to become common in these groups. This gap in understanding is where science ends and speculation begins. Over time, Rh-negative blood became entangled with stories of ancient lineages and hidden ancestry. Some pointed to interbreeding with Neanderthals, suggesting that Rh-negative might be a legacy of those vanished humans who once shared Europe with our ancestors. Others went further, invoking lost civilizations, biblical figures, or beings described in myth as neither fully human nor fully divine. These ideas persist not because they are supported by evidence, but because the mystery feels too large for a simple answer. A single missing protein does not feel like enough to explain so much death, so much pattern, so much cultural memory. Humans have always filled explanatory gaps with story, and Rh-negative blood, rare and consequential, offers fertile ground. But before we follow those stories too far, we need to slow down. We need to step back into the historical moment when this blood factor first revealed its deadly cost, not in legend, but in hospital wards filled with grief. Because the true weight of Rh-negative blood is not in ancient myth. It is in the quiet tragedies that unfolded in the modern world, long before anyone spoke of aliens or lost kingdoms. In the decades before Rh immune globulin was developed, obstetric wards saw patterns that haunted physicians. First, babies healthy. Second, pregnancies complicated. Third and fourth, increasingly dangerous. Exchange transfusions performed on newborns, replacing nearly all of their blood in desperate attempts to save them. Some survived, many did not. These were not rare cases. In regions with high Rh-negative frequencies, thousands of families were affected every generation. Parish records, hospital logs, and family histories all bear traces of this invisible war between mother and child. And still, Rh-negative blood persisted. This is where part one of our journey pauses. We have seen the outline of the mystery, a blood type that should not exist in its current form. A trait that defies simple evolutionary logic, a pattern etched into geography, language, and loss. For centuries, power in Europe flowed through bloodlines. Thrones were inherited, not earned. Authority was justified not by merit, but by ancestry. To rule was to be born correctly, and to be born correctly meant preserving blood, guarding it, narrowing it, keeping it within carefully defined boundaries. Marriage was not romance, it was strategy. Cousins married cousins, nieces married uncles. Dynasties folded back in on themselves again and again like a river forced into a looping channel. No one involved understood genetics. No one knew about recessive traits, immune incompatibilities, or antigen systems. But they believed with absolute conviction that blood mattered, that something essential, something intangible yet powerful, flowed through their veins and must not be diluted. They called it blue blood. The phrase began as a visual metaphor. Pale skin made veins more visible, and among Spanish nobility it became a marker of separation from darker-skinned populations they claimed not to have mixed with. But over time, blue blood became more than appearance. It became identity, destiny, proof of difference, proof of right. What those families did not realize was that by narrowing their bloodlines, they were performing an uncontrolled genetic experiment, one that would expose the quiet mathematics of inheritance in the most brutal way possible. Rare traits do not stay rare in small populations, they concentrate. Hemophilia is one of the most famous examples. A single mutation on the X chromosome, harmless in female carriers, lethal in male heirs. When Queen Victoria unknowingly carried that mutation, it spread outward through Europe's royal houses like an invisible fuse. Sons bled to death from minor injuries. Dynasties trembled because a future king could not survive a fall from a horse or a childhood illness. But hemophilia was not alone. Alongside it, other rare traits clustered: metabolic disorders, skeletal abnormalities, infertility, and quietly unnoticed by history books for decades, Rh-negative blood. Unlike hemophilia, Rh-negative status does not announce itself at birth. It does not deform the body or weaken muscles. A Rh-negative child grows normally, lives normally, marries normally. The danger only appears later and only under specific circumstances. A woman with Rh-negative blood, a partner with Rh-positive blood, a pregnancy, then another. Royal families were particularly vulnerable to this pattern because they valued prolific reproduction. Heirs mattered. Spares mattered. A dynasty was only secure if it produced multiple surviving sons. And so queens and princesses became pregnant again and again, often with little spacing between births. In an era before Rh incompatibility was understood, this was a recipe for disaster. Records from European courts are filled with strange repetitions: firstborn healthy, second sickly, third stillborn, repeated miscarriages, infant deaths blamed on weak constitution, divine will, or moral failing. Midwives whispered, physicians shrugged. The mother was often blamed, her body, her temperament, her supposed fragility. In reality, her immune system may have been doing exactly what it evolved to do. If Rh-negative blood was present in royal lineages, even at modest frequency, its effects would have been magnified by inbreeding and repeated pregnancies. A queen who lost several children could destabilize a nation. Succession crises triggered wars. Wars reshaped borders. All from molecular interactions no one could see. This is not speculation for drama's sake. It is how genetics behaves when constrained by rigid social systems. The same logic applies to any small, closed population, whether royal courts, isolated villages, or religious sects that forbid marriage outside the group. Blood does not care about crowns, it obeys probability. The irony is sharp. Families obsessed with purity believed they were preserving strength. Reality, they were concentrating vulnerability. And this pattern did not end with royalty. It extended into rural Europe, where isolation and tradition kept populations small and intermarriage common. Mountain communities, coastal villages, places where people married neighbors because there was no one else. Here too, Rh-negative blood could quietly accumulate, not because it was chosen, but because it was never diluted. For most families, the consequences were personal, not political. A woman who buried more children than she raised, a household haunted by cradles that never filled, stories passed down without explanation. The second baby never lived, the third was sickly. Something wrong with our women. These narratives echo through genealogy records, church registries, and oral histories. Only in hindsight do they align into a recognizable pattern. By the early 20th century, doctors were desperate for answers. Obstetrics was advancing. Anatomy was mapped. Germ theory was accepted, yet this one phenomenon remained stubbornly mysterious. Why did some mothers lose successive babies while others did not? Why did the pattern repeat so predictably? When the Rh factor was finally identified, it was like turning on a light in a dark room. Suddenly, decades of confusion snapped into focus. Blood transfusion reactions, newborn anemia, pregnancy loss, all connected by a single missing protein. But understanding the mechanism did not immediately solve the problem. For nearly 3 decades after the Rh factor's discovery, doctors could diagnose the risk but not prevent it. They could tell a woman she was Rh negative. They could tell her that future pregnancies were dangerous, but they could not stop her immune system from remembering. Imagine what that knowledge meant in the 1940s and 1950s. A woman told that her first child would likely be fine, but the next might die, that each pregnancy increased the risk, that there was nothing to be done. No injection, no therapy, just probabilities and prayers. Some women chose to stop having children. Others continued, hoping they would be lucky. Many were not. This period, more than any other, reveals the true cost of Rh negative blood, not in myth or theory, but in grief. Hospitals filled with infants undergoing exchange transfusions, their entire blood volume replaced in desperate attempts to save them. Some survived with brain damage, others slipped away while doctors stood helpless. And still, evolution did not erase the trait. This is the paradox that refuses to go away. Natural selection is unforgiving. Traits that significantly reduce reproductive success usually vanish quickly, yet Rh negative blood did not. It persisted through ice ages, migrations, famines, plagues, and centuries of childbirth without modern medicine. Why? One answer is uncomfortable in its simplicity. Chance. In small populations, chance can overpower selection. If a group is isolated enough, and if the reproductive cost is delayed rather than immediate, a trait can persist far longer than logic suggests, especially if social structures like monogamy and inheritance limit mating patterns. But chance alone may not tell the whole story. Some researchers have suggested that Rh negative blood may confer subtle advantages that are difficult to detect. Not dramatic superpowers, not immunity to death, but small shifts, slightly different immune responses, altered susceptibility to certain pathogens, minor changes in how the body handles stress or infection. These effects would not be obvious in individuals. They would only emerge statistically over thousands of years in populations exposed to specific environments or diseases. Consider prehistoric Europe, Ice Age conditions, dense winters, close living quarters, zoonotic diseases spreading from animals to humans. In such environments, even a marginal difference in immune response could mean survival or death. If Rh negative blood altered immune signaling in a way that helped resist a particular pathogen even slightly, that advantage could offset reproductive losses. Not completely, but enough to keep the trait alive. We do not know if this happened. The pathogens of the Paleolithic are gone. Their DNA is rarely preserved. Their selective pressures are invisible to us now. All we see are the survivors. Another possibility is that Rh negative blood is not a single story, but many. The RHD gene can be disrupted in multiple ways. Different deletions, different mutations, some may be ancient, others more recent. What we call Rh negative may be a category that hides several evolutionary paths. In this view, the trait did not arise once and spread. It arose repeatedly in different places under different conditions. Some versions persisted, others vanished. The modern distribution we see is a patchwork of these histories layered over migration and mixing. This explanation fits modern genetic data better than any single origin theory. It removes the need for lost civilizations or non-human ancestry. It accepts complexity rather than mystery, but it does not satisfy everyone because Rh negative blood is not just a genetic variant, it is an experience. People who discover they are Rh negative often learn about it in emotionally charged moments: pregnancy, emergency transfusion, medical warning. The information arrives not as trivia but as risk. Risk shapes identity. When someone is told their blood could attack their own child or that they must never receive Rh-positive blood, the trait becomes personal. It feels consequential. It feels dangerous. And when that person later learns their blood type is rare, unevenly distributed, and historically misunderstood, it is easy to see why the imagination fills in the gaps. Stories emerge, stories about ancient lineages, about being different for a reason, about carrying something old. In the late 20th century, as genetics entered popular culture, these stories exploded. Books, forums, documentaries, and late-night radio shows framed Rh-negative blood as evidence of hidden ancestry. Neanderthal hybrids, biblical figures, visitors from the sky. Theories stacked upon theories. Science pushed back, but not always effectively. Because science tends to say we don't know yet, while myth says here is the answer. And when the question touches identity, loss, and meaning, certainty is seductive. Yet the true story, stripped of embellishment, is already extraordinary. A single protein, absent in a minority of humans, able to turn pregnancy into an immune conflict, able to dictate transfusion compatibility, distributed across the globe in patterns that map ancient migrations and isolations, preserved through millennia of suffering and survival. This is not mundane. And it does not require aliens to be fascinating. By the late 1960s, the story took a dramatic turn. For the first time in human history, medicine found a way to intervene. Rh immune globulin, a simple injection, antibodies given at the right moment to prevent the mother's immune system from ever learning to recognize the Rh protein. The effect was immediate and staggering. Infant deaths from hemolytic disease plummeted. Families that would have lost multiple children now raised them without incident. A problem that had haunted humanity for tens of thousands of years was neutralized in a single generation. From an evolutionary perspective, this was unprecedented. A trait that had imposed a reproductive cost was suddenly freed from it. Rh-negative blood no longer reduced fertility. Natural selection's pressure was lifted. What happens to a genetic trait when its cost disappears? It persists. It spreads according to mating patterns, not survival. Modern medicine does not just save lives, it reshapes evolution. Today, Rh-negative blood is safer to carry than it has ever been. Pregnancy risk is managed, transfusions are controlled. The trait that once killed silently now sits quietly in lab reports and donor cards, but the mystery did not vanish with the danger. If anything, it grew stronger. Freed from fear, people began to ask different questions. Not will this kill my child, but what does this mean about me? By the time modern medicine removed the greatest danger of Rh-negative blood, something unexpected happened. The fear receded, but the curiosity intensified. What had once been a quiet medical hazard became, for many people, a question of identity. If this blood had shaped so much suffering, if it had survived against evolutionary odds, then perhaps it meant something more than science was willing to say. This is how biology becomes belief. Most people discover their blood type casually. A form, a donation card, a routine test, but Rh negative status often arrives differently. It appears during pregnancy when stakes are already high, or in emergency medicine when compatibility means life or death, or in a conversation with a doctor who pauses slightly longer than usual before explaining what the minus sign means. That pause matters. It tells the listener, even subconsciously, that this information is not trivial. And when something feels important before it is fully understood, the mind begins to search for meaning. In the late 20th century, as genetics entered public awareness, people began to talk about blood differently. DNA was no longer abstract, it was ancestry, identity, destiny. Commercial testing companies promised to reveal who you really were. Television programs spoke of genetic memory. Ancient migrations were reconstructed with colorful maps and confident narrators. Rh negative blood slipped neatly into this cultural moment. It was rare, it was consequential, it had a story, and crucially, it did not yet have a satisfying explanation that fit into a single sentence. This is where claims began to grow, not all at once, but gradually, like vines finding cracks in stone. Some people noticed patterns in themselves, sensitivity to light, feeling cold easily. Strong emotional responses, a sense of being different from those around them. These experiences are common across humanity, but when someone learns they are Rh negative, coincidence can transform into correlation. Online forums amplified this effect. People shared lists, traits associated with Rh negative blood, high empathy, strong intuition, vivid dreams, feeling overwhelmed in crowds, a sense of not belonging. Each list differed slightly, but they overlapped just enough to feel convincing. When someone reads a description that partially matches their inner life, it feels like recognition. When they learn that the description applies to a small, rare group, it feels like revelation. This is not deception. It is pattern recognition paired with narrative hunger. Psychologists have long understood this phenomenon. Humans are remarkably good at finding meaning, even when none is intended. Give a group of people vague but flattering descriptions, and many will feel personally seen. Add rarity and the effect intensifies. Add a biological marker and the story gains legitimacy. Rh negative blood became a canvas. Some claims stayed close to physiology. Lower blood pressure, different immune responses, slight variations in disease susceptibility. These ideas are not entirely unfounded. Blood groups do influence how the body interacts with pathogens. The ABO system affects clotting and cardiovascular risk. Rh status has been linked, tentatively, to differences in reaction time and parasite response in limited studies. But tentative findings rarely survive intact in the wild. Nuance does not travel well online. Small statistical differences became absolute statements. Rh negatives don't get this disease. Rh negatives are resistant to that virus. Rh negatives react differently to anesthesia. A grain of data turned into a pillar of belief. Then came the leap. If the body is different, perhaps the mind is too. This is where the narrative crosses from biology into mythology. Claims of heightened intuition appeared first, a sense of knowing, being able to read people easily, feeling energies. These experiences are deeply human and widely reported across cultures, but when attached to a rare biological trait, they feel less subjective, less personal, more inherited. Psychic ability followed. Empathy became telepathy. Pattern recognition became foresight. Dreams became premonitions. The language shifted subtly, but the effect was profound. Rh negative blood was no longer just different, it was special. From there, the door opened fully. Ancient bloodlines, survivors of lost civilizations, Neanderthal hybrids, biblical figures, visitors from beyond Earth. Each theory offered something slightly different, but they all answered the same emotional question: why do I feel the way I do? Science, in contrast, offered uncertainty, maybe chance. Maybe drift, maybe subtle effects we haven't detected yet. That answer is honest, but it is not comforting. It does not elevate difference into purpose, and so belief flourished. It is important to understand why these ideas persist, because dismissing them outright misses the deeper truth. People are not drawn to these stories because they want to feel superior. Most are drawn because they have felt out of place, different, sensitive, out of sync with their surroundings. Rh negative blood offers a language for that feeling. I'm not broken, the story says. I'm rare. There is relief in that, but relief can harden into identity, and identity can become exclusion. Some communities began to treat Rh negative status as a dividing line, us and them, those who know and those who don't, chosen and ordinary. The bloodline language that once justified monarchy quietly resurfaced in a new form. This is where caution is needed. History has seen what happens when biology is used to sort humans into hierarchies. Even when the intention is personal meaning rather than power, the outcome can drift toward separation. Science has tried to address these claims directly. Large-scale studies have looked for links between Rh status and personality traits, intelligence, emotional sensitivity, and cognitive style. When properly controlled for culture, education, and environment, the effects mostly disappear. Any differences that remain are small, inconsistent, and not predictive at the individual level. In other words, your blood type does not determine your soul. But that does not mean the story ends there, because while science has not confirmed psychic powers or ancient ancestry tied to Rh negative blood, it has also not finished exploring its subtler effects. Immunology is complex. The immune system interacts with the brain more than we once believed. Inflammation influences mood. Infection history shapes neural development. Tiny differences can ripple outward over a lifetime. The honest position is not that Rh negative blood means nothing, nor that it means everything. It is that we do not yet know all that it means, and that uncertainty is fertile ground. Interestingly, this is not the only culture where blood has been used to explain personality. In Japan, blood type personality theory focuses on the Abo system rather than the Rh factor. There, people casually associate blood types with temperament. Type A is earnest and anxious. Type B is creative and unpredictable. Type O is confident and assertive. Type AB is complex and mysterious. These ideas are deeply embedded in pop culture despite having little scientific support. They influence dating, hiring, and social expectations. And yet, most people who reference them do so playfully, as a kind of social shorthand rather than a rigid belief system. What's striking is how different cultures choose different parts of the same biological system to mythologize. In Japan, it's Abo. In Western fringe communities, it's Rh negative. The blood is the same. The stories are different. This tells us something important. The meaning does not come from the molecule. It comes from the cultural moment. Rh negative blood entered public awareness during a time of identity upheaval. Traditional narratives were breaking down. Religion was losing authority in some places. Science was advancing faster than people could emotionally integrate. People were searching for ways to understand themselves that felt both personal and ancient. Rh negative blood offered that bridge. It was modern and old, scientific and mysterious, measurable and invisible. And yet, amid all this storytelling, one practical reality remained constant. Rh-negative blood is invaluable in emergencies. In trauma rooms, when there is no time to type and cross-match, doctors reach for O-negative blood. It is the closest thing to universally safe. This has nothing to do with mysticism. It has everything to do with immunology. Fewer antigens mean fewer reactions. As a result, Rh-negative donors, especially O-negative donors, carry a disproportionate medical importance. Their blood is stocked carefully, used sparingly, requested urgently. In mass casualty events, it can mean the difference between chaos and control. This is a quiet form of significance, not glamorous, not mythic, but real. Rh-negative individuals also face practical limitations. They cannot safely receive Rh-positive blood without risk of sensitization. Women of childbearing age must be especially careful. Their compatibility window is narrower, their margin for error smaller. This, too, shapes experience. To live knowing your biology requires extra caution is different from living without that awareness. It fosters attentiveness, sometimes anxiety, sometimes responsibility, sometimes pride in contributing through donation. These emotional responses are grounded, not fantastical. They arise from real constraints. As medicine advances, Rh-negative blood is gaining new relevance. Researchers studying immune reactions, autoimmune disease, and transplant compatibility use Rh status as one variable among many. During global pandemics, scientists have examined correlations between blood types and infection severity, including Rh factor. The results are modest and often inconclusive, but they reinforce an important point. Blood matters in ways we are still uncovering. And then there is the future: synthetic blood, lab-grown red cells. Antigen-stripped universal donors. These technologies aim to remove compatibility barriers entirely. If successful, they could one day make Rh status less critical in emergencies. But until then, the system depends on human donors, on rare blood types, on people whose biology fits the need. This creates a subtle inversion of the myth. Rh-negative blood is not powerful because it is ancient or alien. It is powerful because it is useful. That usefulness does not elevate one person above another. It binds them into a network of dependency. The trauma victim lives because someone donated. The premature infant survives because the right blood was available. The mother delivers safely because medicine intervened at the right moment. These are not stories of chosen blood. They are stories of cooperation. As we move forward in this journey, it becomes clear that the true tension surrounding Rh-negative blood is not between science and myth. It is between uncertainty and meaning, between what we can measure and what we feel, between explanation and experience. Rh-negative blood sits at that intersection. It is concrete enough to matter and mysterious enough to invite story. Long before blood types were written on charts or whispered in hospital corridors, long before minus signs carried warnings and instructions, the people who carried Rh-negative blood lived in a world where survival itself was uncertain. No explanations, no interventions, only cold, hunger, disease, and the constant pressure to endure one more season. To understand why this blood persisted, we have to go back there. Imagine Europe 20,000 years ago. Ice sheets stretched across the north like frozen oceans. Glaciers carved valleys and erased forests. Much of the continent was uninhabitable, locked under ice or tundra. The people who lived there clustered in refuges: southern France, the Iberian Peninsula, parts of Italy and the Balkans, pockets of land where the climate was harsh but survivable. These refuges were not bustling civilizations. They were small, fragile human networks, a few dozen individuals, sometimes fewer. Every birth mattered, every death reshaped the future of the group. This was a bottleneck. Genetic bottlenecks are moments when populations shrink so dramatically that chance begins to dominate heredity. Traits that would normally remain rare can suddenly become common, not because they are superior, but because the people who carry them survive when others do not. The survivors pass on whatever genes they happen to have. Europe experienced several such bottlenecks: ice ages, volcanic winters, epidemics. Each one narrowed the human pool, sometimes to the brink of extinction. Modern genetic evidence suggests that at one point all humans alive today descended from perhaps only a few thousand individuals. In such conditions, evolution does not behave neatly. It stumbles, it improvises, it keeps what works well enough. Rh-negative blood may have slipped through one of these narrow gates. If a small group of survivors happened to include several Rh-negative individuals, the trait could surge in frequency within that group simply by arithmetic. No advantage required, no selection pressure necessary, just survival and reproduction in isolation. Now imagine that group remaining isolated for thousands of years. Mountain ranges block movement, glaciers cut off valleys, rivers shift course. Other populations move around them, not through them. Over time, their genetic signature becomes distinct, not because they are special, but because they are alone. This scenario fits the Basque region almost too well. During the last glacial maximum, the Iberian Peninsula was one of Europe's primary refuges. Human populations retreated there as ice advanced. When the glaciers finally receded, people spread northward again, repopulating Europe, but not everyone moved. Some stayed, some remained in the mountains, some kept their language. Some kept their genes. Rh-negative blood could be one of those preserved traits. But this explanation, elegant as it is, still leaves a question unanswered. Why didn't the reproductive cost erase it later? Once populations grew again, once groups expanded and intermingled, selection should have resumed its usual role. Rh-negative mothers losing children should have had fewer descendants. Over generations, the trait should have declined, unless something counterbalanced the loss. One possibility lies in disease. In prehistoric societies, infectious disease was one of the strongest selective forces. Pathogens evolve quickly. Immune systems respond in complex ways. Small differences in immune signaling can have outsized effects on survival. The Rh factor is not just a transfusion issue. It is part of a broader membrane protein system involved in transporting molecules across red blood cells. Changes to this system may subtly alter how cells interact with pathogens or how the immune system recognizes infected cells. Modern studies have hinted at differences in how Rh-negative and Rh-positive individuals respond to certain infections. The data is inconsistent, often limited by sample size, but the pattern is intriguing enough to keep researchers interested. If in the deep past Rh-negative blood conferred even a slight resistance to a common deadly disease, that advantage could outweigh pregnancy losses, especially in societies where many children died young regardless of blood type. In such a world, survival to adulthood mattered more than total fertility. A woman who lost one or two infants but survived repeated epidemics might leave more descendants than one who bore many children but succumbed to disease. We may never identify the pathogen involved if there was one. It may be extinct, it may have evolved beyond recognition, but its selective shadow could remain, written into the distribution of blood types. Another possibility lies not in biology but in behavior. Traits do not exist in isolation, they cluster. Genes linked on chromosomes travel together. A deletion in one region can hitchhike alongside variations that affect temperament, metabolism, or stress response. If Rh negative status tended to co-occur with other traits that improved survival under certain conditions, the whole package could persist even if one component was costly. For example, heightened stress sensitivity might be maladaptive in modern life, but in a dangerous environment it could increase vigilance. Faster reaction times, greater awareness of threats. Such traits do not require psychic explanations. They emerge from neurobiology shaped by selection. This does not mean Rh negative people today are inherently more sensitive or intuitive. It means that in certain ancestral contexts, genetic combinations associated with Rh negative blood may have mattered. Over time, as environments changed, the advantage faded, but the gene remained. This is how evolution often works. It leaves behind relics, traits that no longer serve their original purpose but persist because they are not harmful enough to remove. There is another angle that complicates the story further: Neanderthals. When modern humans entered Europe, they did not arrive in an empty land. Neanderthals had lived there for hundreds of thousands of years. They were adapted to cold climates, robust, skilled hunters, social, intelligent. For thousands of years, the two populations overlapped. They competed, they avoided, they exchanged ideas, and they interbred. We know this because Neanderthal DNA survives in modern humans, especially those of European and Asian descent. It influences immune response, skin physiology, and metabolism. Some Neanderthal genes were beneficial, others increased disease risk. Natural selection trimmed and reshaped this genetic inheritance over time. Could Rh negative blood be part of that legacy? The answer, based on current evidence, is uncertain but leaning toward no. Neanderthals likely had their own Rh-like systems, but there is no clear indication that Rh negativity specifically originated from them. However, this does not rule out indirect effects. Interbreeding created novel genetic combinations. Some of those combinations may have disrupted existing systems in unexpected ways. A deletion in the Rhd gene could have occurred in a population already carrying Neanderthal-derived variants that altered immune or metabolic pathways. The combination might have behaved differently than either alone. Evolution is not additive, it is interactive. So while Rh negative blood is not Neanderthal blood in any simple sense, the context in which it emerged was shaped by human-Neanderthal interaction. That context matters. What we can say with confidence is this: Rh negative blood is not a mistake. It is a byproduct of survival under extreme conditions, shaped by chance, environment, and history. It is easy, looking back with modern knowledge, to see the pregnancy risk and assume that the trait should never have survived. But this assumes a level of reproductive control and medical awareness that did not exist. In prehistoric societies, many pregnancies failed for many reasons: infection, malnutrition, trauma, exposure. Blood incompatibility would have been one factor among many, not necessarily the dominant one. Selection does not eliminate traits in theory. It eliminates individuals in practice. And in a world where mortality was high across the board, Rh negative blood may not have stood out as especially disadvantageous. This perspective strips the story of mysticism, but not of wonder. A tiny molecular absence survived tens of thousands of years of chaos. It passed through ice ages, migrations, wars, plagues, and cultural revolutions. It shaped lives without being named. It influenced survival without being understood. Only recently did we learn how to stop it from killing, and only recently did we begin to ask what it might have meant before that. As genetic science advances, ancient DNA is beginning to fill in some gaps. Researchers can now extract genetic material from bones tens of thousands of years old. They can identify blood group markers. They can trace population movements with increasing precision. So far, these studies suggest that Rh-negative variants appeared after humans left Africa and diversified in Eurasia. They support the idea of multiple origins rather than a single ancestral event. They reinforce the role of isolation and drift, but the data is still sparse. Ancient remains are rare. Preservation is uneven. Each new genome adds clarity but also complexity. What remains consistent is the pattern: Western Europe, Atlantic fringes, mountain refuges. Places where old things survive. This includes not only blood types but languages, myths, and cultural memory. Humans remember in stories what they forget in facts. The Basque language survived not because it was superior, but because it was protected by geography and continuity. Celtic myths survived on the fringes of empires. Folk traditions persisted where centralized power was weakest. Rh-negative blood may be another survivor of that same process. It is not evidence of lost worlds or non-human ancestry. It is evidence of endurance, and endurance does not always look neat. As we move closer to the present, this endurance takes on new forms. Modern medicine changes selection. Globalization dissolves isolation. Blood types mix freely in cities where people from every continent live side by side. Rh-negative blood will not vanish. If anything, its frequency may slowly increase as medical care removes its reproductive cost. Evolution has entered a new phase, one shaped by technology rather than climate. This raises uncomfortable questions. What traits are we preserving now that would not survive without intervention? What does it mean to alter selection deliberately? Are we accumulating future vulnerabilities we do not yet understand? Rh-negative blood is one of the first clear examples of medicine reshaping evolutionary pressure on a global scale. It will not be the last. And yet, despite all this, the human tendency to mythologize persists. Because even when science explains how something happened, it does not always satisfy why we care. Rh-negative blood matters because it touches life at its most fragile points: birth, death, survival, belonging. It carries history not just in genes but in memory. There is a moment in every mystery when the facts stop speaking loudly enough on their own, and the human mind steps in to fill the silence. Rh-negative blood reached that moment sometime in the late 20th century, when the danger had been tamed but the explanation still felt incomplete. Once medicine removed the immediate fear, curiosity rushed in to take its place. And curiosity, left unguided, often turns inward. People began not by asking where Rh-negative blood came from, but by asking what it said about them. Not as a population, not as a species, but as individuals. Why do I feel different? Why do I think this way? Why have I always felt slightly out of step with the world around me? The answers they found were rarely scientific. They were personal, emotional, interpretive. Someone noticed they were sensitive to noise. Another felt overwhelmed in crowds. Someone else had vivid dreams or strong gut instincts. These experiences are common, scattered throughout humanity, but once attached to a rare biological marker, they gained coherence. They became traits, then patterns, then identity. Lists began circulating: Rh-negative characteristics: empathy, creativity, intelligence, intuition, sensitivity to light and temperature, a strong sense of justice, difficulty fitting into rigid systems, feeling like an outsider even among family. These lists were rarely sourced. They didn't need to be. They resonated, and resonance is powerful. When someone reads a description that mirrors how they've felt their whole life, it creates relief. It says there's a reason. It transforms confusion into explanation, difference into meaning. This is how myths are born in modern times, not from ignorance, but from interpretation layered on uncertainty. Psychologists have long studied this process. Humans are storytelling creatures. We do not experience life as raw data. We experience it as narrative. When something about us feels unusual, we look for stories that make it feel intentional rather than accidental. Rh-negative blood offered such a story. It was measurable, it was inherited, it was rare, it had real consequences, and crucially, it was invisible. You could carry it your entire life without anyone knowing unless you told them. That invisibility made it feel secret, almost sacred, a quiet marker of difference. From there, the leap to deeper meaning was short. Some people framed Rh-negative blood as a survival lineage, descendants of ancient humans who endured ice ages and bottlenecks. Others leaned into spiritual interpretations, a more sensitive nervous system, greater emotional awareness, a heightened connection to others, and then inevitably the boundary between metaphor and literal belief blurred. Ancient bloodlines became literal ancestors. Sensitivity became psychic ability. Intuition became foresight. And somewhere along the way, biology turned into destiny. Claims grew bolder. Rh-negative people were said to be immune to certain diseases, resistant to viruses, less susceptible to parasites, more compatible with nature, less compatible with modern society. The contradictions didn't matter. What mattered was that the story explained discomfort. Science tried to respond, but science speaks slowly. Large studies failed to find meaningful links between Rh status and personality traits. When differences appeared, they were small, inconsistent, and heavily influenced by culture and environment. Blood type explained almost nothing about who someone was as a person. But that message struggled to land, because for someone who feels different, almost nothing feels dismissive. It does not address the lived experience. It does not explain the feeling of being tuned differently, even if that tuning comes from upbringing, temperament, or trauma rather than blood. So the stories persisted. It is important to understand that belief in special traits is not inherently harmful. People seek frameworks that help them understand themselves. The danger arises when belief hardens into hierarchy, when Rh-negative becomes not just different but better. This shift happens subtly. First comes rarity, then significance, then superiority. Not always overt, often implied. We think differently. We feel more deeply. We see things others don't. History has seen this pattern before. Any trait once framed as rare and meaningful can become a boundary marker. The human tendency to form in groups and out-groups does the rest. Ironically, this mirrors the same thinking that once surrounded royal bloodlines, the belief that something intrinsic, invisible, and inherited separated rulers from subjects, that their blood carried qualities others lacked. In both cases, blood becomes a symbol rather than a substance. Science does not deny that blood matters. It matters profoundly. It determines transfusion safety, pregnancy risk, disease interactions. But science also insists on scale, on proportionality. A single antigen does not define a person. Yet even as scientists cautioned against overinterpretation, new layers of intrigue appeared. The immune system and the brain are more connected than previously believed. Inflammation influences mood. Immune signaling affects neural development. Differences in immune genes can shape how individuals respond to stress and infection. This opened a small door. Could Rh status influence neurobiology indirectly, not in a mystical sense but through subtle physiological pathways? Could small differences, amplified over a lifetime, influence perception or behavior? The honest answer is that we don't fully know. Most studies so far suggest that if such effects exist, they are modest. They do not create categories of people. They do not override environment, culture, or personal history. But the absence of strong evidence is not the same as evidence of absence, and this uncertainty keeps the conversation alive. Meanwhile, modern society adds fuel: isolation, disconnection, rapid change. Many people feel misaligned with the pace and structure of contemporary life. When someone who feels overwhelmed by noise, screens, and crowds discovers they are Rh negative, the story writes itself. This world wasn't built for us. That phrase appears often in Rh negative communities. Sometimes explicitly, sometimes implied, it reframes discomfort as misfit rather than weakness, as evolutionary lag rather than personal struggle. There is comfort in that, but comfort does not require literal truth to be effective. Metaphors can heal without being biologically accurate. Problems arise when metaphor is mistaken for mechanism. At the extreme edge of the narrative spectrum lie theories that attempt to explain Rh negative blood through external origins. Not just ancient humans, but non-human intervention, biblical beings, extraterrestrials, lost civilizations with advanced genetics. These theories persist not because of evidence, but because they offer closure. They replace uncertainty with explanation. They answer not only where Rh negative blood came from, but why it feels meaningful. Science rarely offers that kind of narrative satisfaction. Science says, this happened because of mutation, drift, selection, and chance. It does not say this happened for you, and so the mind fills the gap. It is worth pausing here to recognize something important. The human impulse to find meaning in biology is not foolish, it is ancient. Long before science, people looked to the body for signs, birthmarks, eye color. Physical traits were read as omens, destinies, connections to the divine. Rh negative blood is simply a modern canvas for that ancient habit. The difference is that today, the canvas is validated by lab tests and medical charts. It feels objective. It feels real in a way myth alone does not, but objectivity can be misused. This is why it matters to hold two truths at once. Rh negative blood is genuinely unusual in its distribution and consequences, and it does not grant special powers, moral insight, or destiny. These truths are not in conflict. The risk lies in letting mystery become mythology without boundaries. Because when belief shifts from "this helps me understand myself" to "this explains why I am superior or separate," the ground becomes unstable. History shows where that road can lead, yet dismissing all meaning outright is also a mistake. People's experiences are real even if the explanations they adopt are flawed. A person who feels overwhelmed by stimuli may indeed have a more sensitive nervous system. A person who feels emotionally attuned may indeed have high empathy. These traits exist. They just do not belong to a blood type. Understanding that distinction matters. As research continues, we may uncover subtle links between immune genetics and cognition. We may discover that Rh status plays a small role in certain conditions or responses, but those findings, if they emerge, will add nuance, not destiny. They will not turn blood into prophecy. In the present moment, Rh negative blood occupies a strange position. It is medically ordinary and symbolically charged. It is both deeply understood and persistently mysterious. It touches the most intimate aspects of life without offering a clear narrative of why, and that tension keeps people talking. In the modern world, the mystery of Rh negative blood no longer announces itself through loss at birth or unexplained tragedy in family records. Instead, it waits quietly in refrigerators, on shelves behind locked doors, labeled with dates and barcodes, monitored with almost obsessive care. It appears not in myth but in protocol, not in legend but in emergency. Picture a trauma bay at 3 in the morning, fluorescent lights, a stretcher rolling fast, blood pressure dropping. There is no time to ask questions, no time to wait for full laboratory typing. The body on the table is losing blood faster than it can be replaced. In moments like this, medicine does not search for meaning, it searches for compatibility. This is where Rh-negative blood becomes indispensable. In emergencies, doctors reach for blood that is least likely to trigger a catastrophic immune reaction. Blood without A antigens, without B antigens, without the Rhd antigen, O negative, the simplest surface. The least suspicious stranger the immune system is likely to tolerate. This is not mysticism, it is immunology under pressure. The red blood cells do not care who the patient is. They only care what markers they carry and what antibodies are waiting to attack them. In this stripped-down biological logic, Rh-negative blood becomes a universal bridge, not perfect, not absolutely safe in every scenario, but safer than any alternative when time is running out. This is why hospitals guard Rh-negative blood so carefully. It is not abundant. In many populations, fewer than 1 in 10 people carry it. Among those, only a fraction are O-negative. Yet demand does not scale down with rarity. Emergencies do not wait for compatible donors to walk through the door. So blood banks plan, they track usage rates, they rotate stock to avoid expiration, they call donors, they ration units. In some hospitals, O-negative blood is treated almost like a strategic reserve, released only when absolutely necessary. To be Rh-negative in this system is to be quietly valuable, not superior, not chosen, but needed. This need creates a different kind of identity, one grounded not in speculation but in responsibility. Many Rh-negative individuals are encouraged to donate regularly. Some receive calls when supplies are low. Some are listed in rare donor registries, especially if their blood lacks additional antigens that could complicate transfusions for highly sensitized patients. For these individuals, their blood type is not a mystery. It is a role, and that role carries weight. Donating blood is not glamorous. It is routine, sometimes uncomfortable, occasionally inconvenient. But for Rh-negative donors, especially O-negative donors, each unit has a higher probability of being used in a life or death situation. Trauma, surgery, neonatal care, massive hemorrhage. This knowledge changes how some people see their bodies, not as vessels of hidden power, but as contributors to a system of survival that extends beyond the self. There's something deeply human about that. And yet, even here, the story remains complicated. Rh-negative individuals are not just donors, they are also patients. Their compatibility window is narrower. They cannot safely receive Rh-positive blood without risking sensitization. For women of childbearing age, this risk carries long-term consequences. A single incompatible transfusion can create antibodies that complicate future pregnancies. This is why Rh-negative patients are often treated with additional caution. Extra checks, extra verification, redundant labeling. Systems built to prevent irreversible mistakes. The margin for error is smaller. In a strange way, this echoes the ancient story. Rh-negative blood has always lived closer to the edge, more constrained, more dependent on context. What has changed is that now, those constraints are managed deliberately rather than suffered blindly. Modern medicine does not erase difference, it contains it. As research advances, Rh-negative blood has become a point of interest beyond transfusion. Immunologists study it as part of the broader puzzle of how the immune system distinguishes self from non-self. Geneticists examine the Rhd gene to understand how deletions and mutations propagate through populations. Epidemiologists look for subtle correlations between Rh status and disease outcomes. During global health crises, these questions intensify. When new pathogens emerge, scientists look everywhere for clues: age, sex, comorbidities, socioeconomic status, and yes, blood type. During the COVID-19 pandemic, early studies suggested that certain blood groups might influence susceptibility or severity. Type O appeared slightly protective, type A slightly vulnerable. Rh negative status showed hints of reduced risk in some datasets. These findings were modest, often inconsistent, frequently overshadowed by stronger factors like age and underlying health, but they reinforced a key idea. Blood is not passive. It participates in how the body responds to threats. Even a small statistical shift can matter at population scale. This does not mean Rh negative blood confers immunity. It does not mean it makes someone safer in any absolute sense, but it suggests that the trait remains biologically relevant, not just historically interesting. And relevance invites attention. Meanwhile, technology pushes forward. Researchers are working to engineer red blood cells in the lab, to grow them from stem cells, to strip them of surface antigens, to create a truly universal donor cell that can circulate safely in any body. These efforts are driven in part by the challenges posed by rare blood types. Patients with complex transfusion histories sometimes develop antibodies against multiple antigens. For them, finding compatible blood can be nearly impossible. A synthetic or lab-grown alternative could save lives. If such technology succeeds, it may one day reduce dependence on human donors altogether. What happens to the meaning of Rh negative blood then? From a logistical standpoint, its importance might diminish. No more shortages, no more urgent calls, no more strategic reserves, but from a scientific standpoint, its importance may increase. Rh negative blood serves as a template, a model of minimal antigenicity, a guide for designing cells the immune system will tolerate. In this way, the trait that once caused suffering becomes a blueprint for safety. There is a quiet symmetry in that. Looking forward, the evolutionary story of Rh negative blood enters a new phase. For tens of thousands of years, its frequency was shaped by chance, isolation, disease, and reproductive risk. Over the last half-century, medicine has intervened, removing its greatest cost. Selection pressure has changed. In evolutionary terms, this is significant. Traits that were once constrained by mortality are now free to persist and spread. This does not mean Rh negative blood will suddenly dominate. It simply means it is no longer being actively pruned by pregnancy loss. The human genome is entering an era where technology reshapes selection more powerfully than environment. Rh negative blood is one of the earliest examples of this shift at a global scale. This raises questions that extend far beyond blood type. What does it mean to preserve traits that once carried risk? How do we balance compassion with unintended consequences? Are we accumulating vulnerabilities that future medicine will need to manage? There are no simple answers, but Rh negative blood reminds us that evolution is not a story with a clear ending. It is an ongoing process, now entangled with ethics, technology, and choice. And still, despite all this complexity, the human tendency to seek meaning remains. Some people will always feel that their Rh negative status explains something essential about who they are. Others will see it as a footnote, a medical detail, a donor card. Neither perspective is wrong as long as it does not harden into exclusion or destiny. Because the deepest truth about Rh-negative blood is not that it makes someone different, it is that it connects them. It connects a mother to a child through antibodies and intervention. It connects a donor to a stranger through transfusion. It connects modern humans to ancient populations through shared inheritance. It connects science to history, medicine to myth. The same red cells that once triggered immune conflict now flow safely through neonates and trauma victims because someone learned how to intervene at the right moment. The same genetic absence that once puzzled evolution now helps guide innovation. This is not a story of failure or anomaly. It is a story of adaptation. As we approach the final stages of this journey, one question remains beneath all others. Not where Rh-negative blood came from, not what powers it might grant. But what it reveals about us. It reveals how fragile survival once was, how easily chance shapes destiny, how much suffering can be hidden in something invisible, and how human ingenuity can transform danger into care. When you step far enough back from the details, from the antigens and antibodies, the charts and hypotheses, a different picture begins to emerge. Not one of mystery in the sense of something supernatural, but of complexity so layered that no single explanation can fully contain it. Rh-negative blood stops being a puzzle to solve and becomes a mirror, reflecting how humans respond to uncertainty when it runs through their own bodies. Because at its core, this story is not really about blood. It is about how we interpret difference. For most of human history, difference was dangerous. To stand out was to risk exclusion. To carry a trait others did not understand invited suspicion. And yet, difference was also how populations survived. Variation is the raw material of adaptation. Without it, extinction comes quietly and quickly. Rh-negative blood embodies this tension perfectly. It is difference with consequence, difference that could kill, difference that could save, difference that survived without asking permission. In prehistoric societies, no one celebrated or condemned Rh-negative blood. They didn't know it existed, they simply lived with its effects, folded into the larger uncertainty of life. Pregnancy was dangerous for everyone, disease was everywhere, survival was uneven, loss was common. In that world, Rh-negative blood was just one thread among many rarely singled out. It was only when humans began to understand themselves scientifically that this thread stood out. Only when we learned to isolate variables did Rh-negative blood become a named thing, a category, a risk factor, a curiosity. And with naming came meaning. Once something is named, it can be talked about. Once it can be talked about, it can be interpreted. Once it can be interpreted, it can be claimed. This is how a medical classification slowly turned into an identity. But identity is never purely biological. It is shaped by context, by timing, by culture. Rh-negative blood might have remained an obscure immunological footnote if it were evenly distributed, harmless, and unremarkable. It became charged because it was rare, uneven, and dangerous under specific conditions. Rarity invites attention. Danger invites story. Consider how many people first learn about their Rh status, often during pregnancy, a moment already heavy with anticipation, fear, hope, and vulnerability. To be told that your body might reject your own child unless medicine intervenes is not a neutral experience. It imprints itself. Later, when that same person encounters stories suggesting their blood is ancient, special, or meaningful, those stories find fertile ground. They attach themselves to memory and emotion, not just intellect. This is why purely factual rebuttals often fail. They address the claim, but not the need the claim is fulfilling. The need is not to believe in aliens or lost civilizations. The need is to understand why something so small could have mattered so much. And the honest answer is that sometimes, in evolution, it doesn't need to mean anything at all. This is one of the hardest truths science offers. Not every trait exists for a reason. Not every outcome is purposeful. Some things persist because nothing strong enough removed them. Humans resist this idea instinctively. We prefer stories with intention, with cause and effect that feels proportional. We struggle with the idea that suffering can be accidental, that patterns can emerge without design. Rh-negative blood challenges that preference. It asks us to accept that a deletion in a gene occurring by chance in a small population could echo across tens of thousands of years, that it could shape birth and death without ever being meant to do so. That is a deeply unsettling idea, and yet it is also strangely grounding. Because if Rh-negative blood teaches us anything, it is that humans are not engineered for perfection. We are assembled through survival, through compromise, through accidents that worked well enough to continue. The same genetic processes that produced Rh-negative blood also produced lactose tolerance, sickle cell trait, and countless other variations that are beneficial in some contexts and harmful in others. Evolution does not optimize globally, it patches locally. This is why the same trait can be a liability in one era and neutral or even useful in another. Before modern medicine, Rh-negative blood carried risk. After the development of Rh immune globulin, that risk largely disappeared. The trait did not change, the environment did. This shift alone should give us pause when assigning deep meaning to biology. What feels like destiny in one context can become trivial in another. What feels like a curse can become a footnote. What feels like a mystery can become routine. And yet, even as Rh-negative blood becomes medically manageable, the questions do not vanish, they evolve. Now we ask not how to survive it, but what it reveals. It reveals how fragile reproduction once was, how many losses were absorbed into family histories without explanation. How many women blamed themselves for something written in molecules they could not see. It reveals how deeply interconnected humans are. A donor in one city saves a stranger they will never meet. A genetic variant carried by ancient hunters influences protocols in modern hospitals. It reveals how culture overlays meaning onto biology, sometimes gently, sometimes dangerously. And it reveals how difficult it is to live with uncertainty. Rh-negative blood sits in a category of traits that are understood enough to matter, but not enough to feel complete. Those traits attract speculation the way gravity attracts matter. Not because people are irrational, but because the human mind dislikes unfinished stories. If the story ended with "this blood type exists because of random mutation and drift," many would feel unsatisfied. It sounds too small for the scale of impact, but scale is deceptive. A single missing protein can change how the immune system behaves. A single immune reaction can determine whether a pregnancy succeeds. A single birth can alter a family's future. Enough families and the pattern becomes visible across regions. Small causes, large effects. This is not mystery. It is chaos theory applied to biology. And chaos theory, like evolution, is uncomfortable because it removes intention. Yet within this discomfort is a quieter, more human meaning. Rh-negative blood reminds us that survival has always been collective. No individual trait guarantees endurance. What matters is how systems respond, how groups adapt, how care is shared. The tragedy of Rh incompatibility only ended when humans learned to intervene collectively. When knowledge was pooled, when medicine was distributed, when the fate of one family became a problem worth solving for everyone. That is the real turning point in this story. Not the discovery of the Rh factor itself, but the decision to act on it universally. This is where the narrative shifts from biology to ethics. Modern medicine did not ask whether Rh-negative blood should exist. It asked how to reduce suffering. It did not judge the trait, it accommodated it. This approach stands in contrast to older ways of thinking about difference. Instead of eliminating variation, we manage it. Instead of purifying bloodlines, we protect individuals. This is not evolution's logic, it is humanity's, and it is reshaping the future. As genetic technologies advance, we will face similar questions again and again. Which traits do we correct? Which do we tolerate? Which do we preserve? Which do we reinterpret as difference rather than defect? Rh-negative blood is an early case study in this shift. It shows both the promise and the complexity of intervening in natural processes. It also shows that meaning does not have to come from exceptionalism. You do not need to believe your blood is alien, ancient, or superior for it to matter. It matters because it is part of a network of care, because it once caused harm and now saves lives. Because it connects you to strangers through systems you will never see. There is a quiet dignity in that. If there is any sense in which Rh-negative blood is special, it is not because it sets someone apart, but because it reveals how deeply interdependent humans are, how the survival of one depends on the attention of many. This may not be as thrilling as ancient astronaut theories or secret bloodlines, but it is far more true, and perhaps more meaningful. As we come to the end of this part of the journey, the mystery has not vanished. It has transformed. It no longer asks, "What are you?" It asks, "What do you do with what you are?" Rh-negative blood does not dictate destiny. It presents conditions. How those conditions are met depends on knowledge, culture, and choice. In earlier eras, those conditions led to loss. In the modern era, they lead to protocols, donor lists, and life-saving injections. In the future, they may lead to synthetic blood and entirely new understandings of compatibility. The blood remains the same. The story around it changes, and that may be the most important lesson of all. As we look forward beyond history and speculation, beyond myth and memory, Rh-negative blood begins to feel less like a riddle left behind by evolution and more like a signpost pointing towards something unfinished. Not unfinished in the sense of error, but unfinished in the sense that the story is still unfolding, shaped now not only by biology but by human intention. For the first time in our species' existence, we are not merely subject to evolution, we are participants in it. For hundreds of thousands of years, genetic variation emerged blindly. Mutations occurred, some helped, some harmed, most did nothing at all. The environment decided which survived: ice, hunger, disease, childbirth. There was no appeal, no intervention, no second chance. Rh-negative blood lived through that era. It survived not because it was protected, but because it was tolerated by circumstance. Now that has changed. Modern medicine does something evolution never did. It protects the vulnerable trait without erasing it. It does not select against Rh-negative blood. It buffers it. It wraps it in protocols, injections, safeguards, and logistics. It makes room for difference rather than pruning it away. This is a radical shift. From an evolutionary perspective, Rh-negative blood should have declined in frequency wherever it caused repeated reproductive loss. That pressure is gone now, at least in societies with access to health care. An Rh-negative woman can have multiple Rh-positive children without consequence. Her immune system is gently guided away from conflict. Selection pressure has been altered. This raises a quiet but profound question. What happens when traits once constrained by biology are freed by technology? The answer is not dramatic. There is no sudden explosion of Rh-negative blood. Genetic frequencies change slowly, but the direction changes. Traits that once hovered at the edge of persistence now have room to remain, perhaps even to spread slightly through random mating. This is not unique to Rh-negative blood. It applies to countless conditions once lethal in childhood that are now manageable. Diabetes, congenital heart defects, metabolic disorders. The human gene pool is changing not because we are editing it directly, but because we are changing the rules of survival. Rh-negative blood is one of the earliest and clearest examples of this shift, and it forces us to reconsider what it means to ask whether a trait should exist. That question assumes a natural order with intent. Evolution does not ask what should exist, it only reflects what does. When humans intervene, we introduce values, compassion, preservation, choice. These values do not replace biology, they coexist with it. So when people ask whether Rh-negative blood had a purpose, they are often asking the wrong question. The more revealing question is how humans responded to it once they understood it. They did not attempt to eliminate it. They did not stigmatize it into extinction. They did not restrict reproduction. They learned how to manage it. This response tells us something important about who we are becoming. In the past, difference was something to fear or control. In the present, difference is increasingly something to accommodate. This does not mean all differences are harmless or trivial. It means we are shifting from a survival-based framework to a care-based one. Rh-negative blood illustrates this transition with unusual clarity. It also highlights the limits of purely genetic thinking when applied to identity. As genetic testing becomes more widespread, more people will learn intimate details about their biology. Variants, risks, probabilities, predispositions. Without careful framing, this information can easily be misinterpreted as destiny. Rh-negative blood shows how tempting that misinterpretation can be. A single genetic detail becomes a lens through which a person views their entire life, their personality, their relationships, their sense of belonging. But biology does not work that way. Genes influence tendencies, not narratives. They shape probabilities, not purpose. The danger is not in knowing your biology. The danger is in letting it define your worth. History offers many warnings about this. Bloodline thinking has justified inequality, exclusion, and violence. Even when intentions are benign, even when the story is about being special rather than superior, the structure is the same. Identity rooted in biology becomes rigid. It hardens. Rh-negative blood does not need to carry that burden. Its real lesson is not that some people are different in a meaningful cosmic sense. It is that small variations can have large consequences, and that humans are capable of responding to those consequences with intelligence rather than fear. As we move deeper into the genomic age, we will face more traits like this. Variants that affect health, cognition, emotion, and behavior in subtle ways. Traits that cluster in certain populations. Traits that raise uncomfortable questions about fairness, access, and responsibility. Rh-negative blood offers a template for how to navigate those questions. First, understand the biology clearly. No exaggeration, no mystification, just mechanism. Second, acknowledge the human impact honestly. The suffering, the fear, the lived experience. Third, intervene where possible to reduce harm without erasing difference. Fourth, resist the urge to assign moral or existential meaning to a molecule. This is not easy. Humans are meaning-making creatures. We will always tell stories about ourselves. The goal is not to stop storytelling, but to ground it. In this sense, the most compelling story of Rh-negative blood is not ancient or extraterrestrial. It is modern and human. It is the story of a discovery that turned tragedy into prevention, of a simple injection that spared millions of families, of systems built to protect both mother and child, of donors who quietly sustain strangers, of scientists who chose to ask how instead of why. It is a story of care, and care changes how difference feels. When difference is dangerous and unmanaged, it becomes a source of fear and myth. When difference is understood and supported, it becomes a detail rather than a destiny. Rh-negative blood has crossed that threshold. The mystery that remains is not biological, it is philosophical. What do we do with the knowledge that our bodies carry accidents of history, that some of our most intimate traits exist not because they were chosen, but because they survived? Do we seek to elevate those accidents into identity, or do we integrate them into a broader understanding of shared humanity? There is no single correct answer. People will choose differently. Some will continue to see their Rh-negative status as a meaningful personal symbol. Others will treat it as a medical footnote. Both positions can coexist as long as neither demands hierarchy. What matters is remembering that blood connects more than it separates. The same red cells that carry antigens also carry oxygen. The same circulation that can trigger immune conflict also sustains consciousness. The same biological systems that differentiate us also unite us in vulnerability. When viewed from that perspective, Rh-negative blood becomes less about who you are and more about how intertwined human lives have always been. It connects ancient hunters in glacial refuges to modern clinicians in brightly lit hospitals. It connects mothers who once lost children to mothers who now receive a preventative injection without a second thought. It connects donors to recipients, past to present, biology to ethics, and perhaps that is enough. Perhaps the true meaning of Rh-negative blood is not hidden in prehistory or encoded in destiny, but visible in how a species learns. Learns from suffering, learns from patterns, learns how to intervene without erasing, learns how to hold difference without mythologizing it into separation. If there is a future lesson here, it is this. As our ability to understand and alter biology grows, our responsibility to interpret it wisely grows with it. Rh-negative blood reminds us that understanding without humility leads to myth, and humility without understanding leads to fear. Balance lies in holding both. The blood itself will keep flowing, indifferent to our stories. It will carry oxygen. It will circulate nutrients. It will do what blood has always done. What changes is what we build around it. And that, ultimately, is the part of the story that belongs not to evolution, but to us. It circles back to something deceptively simple, something that has been present from the very beginning but easy to overlook when theories grow large and language grows dramatic. Blood does not know the stories we tell about it. It does not carry intention, prophecy, or identity. It carries oxygen. It carries life. Everything else is layered on top by human minds trying to make sense of what feels personal and profound. Yet those layers matter because humans live inside them. For someone who learns they are Rh-negative, the information rarely arrives alone. It arrives with context, a warning, a protocol, a note of caution. It arrives during pregnancy or crisis or donation, moments when the body is suddenly no longer abstract. It is vulnerable. It is finite. It can fail. That emotional framing shapes interpretation. When people later encounter stories suggesting their blood connects them to ancient survivors, or that it marks them as unusually sensitive or perceptive, those stories resonate not because they are proven, but because they answer a feeling that already exists. A feeling of fragility, of difference, of being slightly misaligned with the world. This is where it becomes important to slow down. Feeling different is not the same as being biologically different in a meaningful categorical way. Human variation is continuous, not divided into hidden castes. Temperament, perception, and sensitivity arise from countless interacting factors: genes, development, environment, experience, trauma, culture. No single marker can claim ownership of them. Rh-negative blood is part of the body, not the self. And yet, dismissing the subjective experience entirely would be a mistake. People are not imagining their failings, they are interpreting them. The problem is not that they feel sensitive, intuitive, or out of place. The problem arises when a biological explanation is retrofitted to validate those feelings as innate destiny rather than lived experience. The distinction matters. One path leads to curiosity and self-understanding. The other leads to rigidity. Throughout history, whenever biology has been turned into identity, the outcome has been double-edged, sometimes empowering, often divisive. Bloodlines have justified hierarchy, purity, exclusion. Even when the story begins as personal meaning, it can drift toward separation. Rh-negative blood does not deserve that weight. It is not a lineage of the chosen. It is not a marker of superiority or inferiority. It is a variation that survived because evolution is messy and medicine intervened. That is enough. There is a temptation to ask whether something this persistent must have a hidden advantage. Perhaps it does. Perhaps not. Biology is full of traits that persist simply because nothing eliminated them. Neutral drift is not dramatic, but it is powerful. Over time, chance accumulates structure. What makes Rh-negative blood feel different is not that it violates evolutionary logic, but that it exposes its rough edges. Pregnancy incompatibility makes the cost visible. Geographic clustering makes history legible. Medicine makes intervention obvious. Most traits do not reveal themselves so clearly. This clarity creates the illusion that a deeper explanation must exist, that if we could just dig far enough, we would find the reason, the purpose, the meaning. But meaning does not live in molecules. Meaning lives in response. The most important chapter in the story of Rh-negative blood is not its origin, but how humans chose to respond once they understood it. They did not attempt to eliminate Rh-negative individuals. They did not restrict reproduction. They did not declare the trait defective. They built systems to protect mothers and children. They designed protocols to ensure safety. They organized donation networks to support emergencies. They adapted socially to a biological reality. This response is not inevitable. History offers many examples of societies choosing exclusion instead. The fact that Rh-negative blood became a problem to solve rather than a trait to eradicate is significant. It suggests a shift in how humanity relates to difference. That shift is still incomplete. It is uneven across regions. Access to medical care is not universal. In some parts of the world, Rh incompatibility still causes preventable harm. The story is not finished, but the direction matters. Rh-negative blood also highlights a quieter truth about modern life. Much of what shapes our survival now happens at levels we never see, in protocols, in supply chains, in systems of coordination. The drama of survival has moved from the wild into infrastructure. A person survives a car accident not because of strength or luck alone, but because someone donated blood months earlier. A newborn survives not because of ancient resilience, but because a clinician followed a checklist and administered an injection. These are not heroic narratives, they are collective ones. And collective stories rarely feel as emotionally satisfying as myths of special origin. But they are more honest. The blood that once marked danger now marks connection. A donor and a recipient linked by compatibility. A mother and a child linked by prevention rather than conflict. A population linked across generations by shared care. This does not erase individuality, it reframes it. Individual experience still matters. Some people will always feel more sensitive, more reflective, more attuned. Others will feel grounded, practical, steady. These differences are real and valuable. They just do not belong to a blood type. When people ask what Rh-negative blood means, a useful answer might be this. It means your body participates in a system that required human ingenuity to make safe. It means you carry a variation that taught us something about immune memory, pregnancy, and care. It means you are part of a story where understanding replaced superstition and prevention replaced tragedy. That is not mystical, but it is profound. As we continue into the future, the lessons of Rh-negative blood will echo in new contexts: gene editing, personalized medicine, predictive genomics. We will increasingly be told what risks we carry, what probabilities shape us, what variants define us statistically. The challenge will be learning how to hold that information without turning it into identity or destiny. Rh-negative blood offers a warning and a guide. The warning is this: when biology is overinterpreted, it becomes mythology. When mythology becomes identity, it can divide. The guide is this: when biology is understood and supported, it becomes just one part of a larger human story. The blood itself will remain indifferent. It will continue circulating, performing its function, oblivious to how it is discussed online or remembered in families. It does not seek explanation. We do. And that seeking says more about us than it ever will about blood. As this narration moves forward, the focus narrows again, not toward origins or futures, but toward perspective, toward the quiet recognition that Rh-negative blood is neither a secret code nor a cosmic accident. It is a reminder of scale, of how small changes ripple outward, of how fragile systems can be and how resilient humans can become when they learn. The mystery, then, is not unresolved because we lack data. It is unresolved because we keep asking it to answer questions it was never meant to address. Who am I? Why do I feel this way? Where do I belong? Those questions cannot be answered by antigens, but they can be informed by the humility that comes from understanding how much of who we are is shaped by chance, history, and care rather than purpose. Rh-negative blood survived not because it was special, but because humanity eventually became capable of protecting it. That may be the most important distinction of all, and with that understanding, the story does not end, it simply quiets. The questions soften, the need for extraordinary explanations fades, replaced by something more sustainable, an appreciation for how ordinary biology, when met with attention and compassion, becomes enough. There's a subtle shift that happens when a mystery has been lived with long enough. At first, it demands answers. Then, over time, it begins to teach restraint. Rh-negative blood has reached that stage. It no longer asks to be solved so much as understood in proportion. And proportion is what humans struggle with most. We are drawn to extremes, to explanations that feel complete, elegant, and emotionally satisfying. We prefer stories where causes match effects in scale. Something that shaped millions of lives must have a grand origin. Something that caused so much pain must have a hidden purpose. Something rare must mean something more. But nature is not obligated to meet our narrative expectations. One of the hardest lessons biology offers is that significance does not require intention. A tiny molecular change can ripple outward through generations without ever being for anything. Rh-negative blood is one such ripple. It is not an anomaly screaming for explanation. It is a quiet consequence of how life actually works. Mutation happens, drift happens, survival happens unevenly. What makes Rh-negative blood feel different is not its origin, but its visibility. We see it in hospital charts, we see it in pregnancy warnings, we see it in blood bank shortages. Its effects intersect with moments of fear, care, and urgency. That intersection amplifies meaning, and humans respond to amplified meaning by telling stories. Some stories are grounded, others drift. But all of them reveal the same underlying impulse, a desire to place ourselves inside something larger than randomness. This impulse is ancient. Long before genetics, people explained birth outcomes through fate, divine will, curses, or blessings. When a baby lived or died, there had to be a reason. When loss repeated itself, the reason had to be big enough to bear the weight of grief. Incompatibility, before it was understood, fit perfectly into this worldview. A woman whose babies died one after another would have been surrounded by explanations that blamed her body, her blood, her ancestry, her morality, or her gods. None of those explanations were true, but they provided structure in a world without answers. Science eventually replaced those stories with mechanism: antibodies, antigens, immune memory, placental transfer. Suddenly the tragedy had a name, but names do not erase emotional residue. They simply change the language it speaks in. Even now, when the risk is controlled in much of the world, the echo remains. The idea that a mother's body could harm her child feels fundamentally wrong. It violates our deepest assumptions about biology and care. That violation lingers in the imagination long after the danger is gone. This lingering discomfort is fertile ground for meaning-making. It is why Rh-negative blood continues to attract attention disproportionate to its biological scope. It touches something primal. It reminds us that bodies are not always aligned with our expectations of them. That nature is not always gentle. That survival sometimes requires intervention. These realizations unsettle us, and so the story expands. It reaches backward into prehistory, searching for an origin dramatic enough to justify the impact. It reaches outward into mythology, borrowing language of chosen lineages and ancient survivors. It reaches inward into psychology, offering identity and explanation to those who feel different. None of this makes the story false. It makes it human, but it does mean we must be careful, because when stories about biology begin to replace biology itself, confusion follows. People start to attribute traits to blood that arise from upbringing or personality. They start to see destiny where there is probability. They start to divide where variation exists. Rh-negative blood becomes a symbol rather than a fact, and symbols are powerful. They shape how people see themselves and others. They can inspire care or justify exclusion. They can comfort or distort. The challenge is not to strip Rh-negative blood of all symbolic meaning, but to keep that meaning tethered to reality. Reality says this: Rh-negative blood is one of many human variations. It is neither rare enough nor impactful enough to define a class of people. It does not correlate strongly with intelligence, intuition, empathy, or spirituality. It does not encode personality. What it does encode is immunological difference. An immunological difference matters in specific contexts: pregnancy, transfusion, disease response. That is where its story lives. Outside those contexts, its influence fades into statistical noise. This does not make the trait uninteresting. It makes it specific. Specificity is something myth resists. Myth prefers universality. If a trait matters, it must matter everywhere all the time. Biology rarely works that way. The Rh factor matters intensely in a narrow window and barely at all elsewhere. That asymmetry feels strange to us. We are not comfortable with things that are sometimes life or death and sometimes irrelevant, so we fill the gaps. As we move further into the future, this pattern will repeat with other genetic findings. People will learn they carry variants associated with risk or resilience. Some will internalize that information cautiously. Others will build identities around it. The story of Rh-negative blood can help us navigate that future more wisely. It shows us what happens when a trait is both biologically real and emotionally charged. It shows how easily explanation becomes exaggeration, and it shows how essential it is to separate care from myth. Care says this trait requires attention in specific situations. Myth says this trait defines who you are. Care adapts systems. Myth reshapes identity. When care leads, outcomes improve. When myth leads, confusion follows. The success of Rh immune globulin is not just a medical achievement, it is a philosophical one. It demonstrates that understanding a mechanism does not require assigning meaning beyond function, that we can intervene effectively without rewriting identity. That is a lesson worth carrying forward, because as genetic knowledge expands, the temptation to rank, label, and sort will grow stronger. The tools that allow us to see variation also allow us to overemphasize it. Rh-negative blood reminds us to resist that impulse. It invites us to ask better questions, not what does this make me, but how does this work, not why am I different, but how do we support difference. Those questions lead to better answers. At the end of this long exploration, what remains is not a solved mystery, but a reframed one. The question is no longer why does Rh-negative blood exist, but what does our response to it say about us? It says we are capable of learning from pain. It says we can replace superstition with understanding. It says we can design systems that protect without erasing. It says we can live with variation without turning it into hierarchy. That may not satisfy the desire for cosmic explanation, but it offers something more sustainable, a way of living with complexity. Rh-negative blood will continue to flow through millions of people quietly, unremarkably, doing what blood has always done. It will not announce itself. It will not demand belief. It will simply exist. What we choose to build around it, stories, systems, meanings, that is where the real narrative lives, and that narrative is still being written. At some point, every long investigation reaches a quiet plateau. The urgency fades, the questions soften, and what remains is perspective. Rh-negative blood, once framed as anomaly, danger, and mystery, settles into this space. Not solved, not dismissed, but understood well enough to stop shouting for explanation. And in that quiet, something clearer emerges. The story was never really about blood types. Blood was simply the entry point. What this journey has traced is the human response to uncertainty when it intersects with the body itself. When something unseen, inherited, and uncontrollable reaches into the most intimate parts of life, birth, survival, identity, that is where meaning rushes in. For early humans, the body was fate. There was no separation between biology and destiny. If a trait caused loss, it was woven into belief. If survival followed, it was attributed to favor. There was no alternative framework. Science disrupted that framework, but it did not erase the emotional residue left behind. It replaced one language with another, but the need for coherence remained. Rh-negative blood sits precisely at that crossroads. It is explained enough to be managed, but not explained enough to feel emotionally complete. It has a mechanism, but not a narrative arc that satisfies the human need for meaning. That incompleteness keeps the story alive. Yet incompleteness is not failure, it is honesty. One of the most difficult things to accept about being human is that not everything that affects us has a reason proportional to its impact. Some things simply happen and then we respond. Evolution does not guarantee fairness. Biology does not ensure comfort. Survival does not imply intention, but response, that is where humanity enters. The response to Rh-negative blood transformed it from a quiet source of tragedy into a manageable condition. From a hidden danger into a known variable, from a cause of grief into a routine part of prenatal care. That transformation did not require myth, it required attention. Attention is underrated, it lacks drama. It does not create heroes or legends, but it changes outcomes. A clinician paying attention to blood type, a researcher paying attention to patterns, a donor paying attention to a phone call. These small acts accumulate into systemic care, and systems matter more than stories. The stories we tell about Rh-negative blood may continue to evolve. Some will remain grounded, others will drift. That is inevitable. Humans will always use biology as a canvas for identity, especially when it feels personal. The goal is not to stop that process, but to keep it flexible. Rigid identities fracture, flexible understanding adapts. If Rh-negative blood teaches anything enduring, it is that adaptability, social, medical, ethical, is more powerful than any single genetic trait. It is adaptability that allowed this variation to persist without devastation. It is adaptability that turned incompatibility into prevention. And adaptability is not encoded in blood, it is learned. As the future unfolds, there will be new discoveries that challenge our assumptions about the body, new variants identified, new correlations proposed, new anxieties sparked. Some of them will matter deeply, others will fade. Rh-negative blood offers a template for navigating those moments. Pause before assigning meaning, separate mechanism from metaphor, address harm without creating hierarchy. Let care lead before identity hardens. This approach does not diminish individuality, it protects it. Because the greatest risk in overinterpreting biology is not that we misunderstand science, but that we misunderstand ourselves. That we reduce complex human lives to single traits, that we mistake variation for division. Rh-negative blood is not a dividing line, it is a reminder of connection. Connection across time as a mutation carried forward through generations. Connection across geography as a trait clustered and spread. Connection across strangers as blood moved from arm to arm. Connection across disciplines as biology met ethics and care. In the end, that may be the most fitting place for this story to rest, not in answers that close the mystery, but in understanding that allows it to coexist with uncertainty. You do not need Rh-negative blood to be extraordinary for it to matter. You do not need it to define you for it to be significant. You do not need a grand origin story for it to have shaped lives. Sometimes the most meaningful truths are also the most ordinary. Blood flows, bodies vary, humans notice, respond, and adapt. That is the pattern. And within that pattern, Rh-negative blood is one thread among many, distinct, consequential in specific moments, and otherwise quietly doing its work. The mystery remains only if we insist it must be more than that. If we allow it to be exactly what it is, a biological variation shaped by chance and softened by care, it becomes something calmer, something grounded, something human, and perhaps that is enough. As we arrive at the end of this long journey, the tone naturally changes, not because the mystery has vanished, but because it no longer needs to shout. What began as a question about a missing protein on a red blood cell has unfolded into something much quieter and much larger, a meditation on how humans live with uncertainty written into their own bodies. Rh-negative blood does not resolve itself into a single answer. It never has, and perhaps that is precisely why it continues to hold attention. It sits at the intersection of biology and belief, science and story, mechanism and meaning. It refuses to belong entirely to any one of them. For a long time, people searched for an origin dramatic enough to justify its impact. ancient survivors, lost civilizations, divine bloodlines, external intervention. These ideas persist not because evidence supports them, but because they satisfy a hunger for narrative symmetry. They make the story feel worthy of the suffering it once caused, but biology is not obligated to be poetic. The real history of Rh-negative blood is not epic in the mythic sense. It is incremental, quiet, accidental, a deletion here, a population bottleneck there, a pattern emerging slowly across thousands of years. No intention, no plan, just survival unfolding unevenly. And then very suddenly, in evolutionary terms, something changed. Humans noticed. They noticed that some pregnancies failed for reasons that repeated themselves. They noticed that some transfusions killed instead of healed. They noticed patterns where there should have been randomness, and instead of accepting those outcomes as fate, they asked how. That question, how instead of why, is where this story truly turns. Once the mechanism was understood, the response followed, not elimination, not segregation, not fear, but prevention, accommodation, care. A small injection that changed millions of lives, a protocol that transformed grief into routine safety. A system that quietly protected future generations without asking them to be different. That response is easy to take for granted now, but it represents something extraordinary. It represents a moment when humanity chose to meet biology not with myth or judgment, but with understanding. Rh-negative blood did not need to be explained away. It needed to be supported, and that may be the most important lesson it offers. In a world increasingly obsessed with identity, classification, and explanation, Rh-negative blood reminds us that not every difference demands interpretation beyond function. Not every variation needs a story bigger than itself. Some things simply require care. That does not mean the curiosity was wrong. Curiosity is how understanding begins, but curiosity untethered from humility can drift into fantasy, and fantasy, when mistaken for biology, can divide. The quiet truth is this: Rh-negative blood is neither a mark of destiny nor an accident that defies logic. It is a normal outcome of how life works, variation arising from chance, shaped by history, softened by human intervention. It does not define personality. It does not grant hidden abilities. It does not separate humanity into meaningful categories. What it does reveal is how fragile life once was, how much suffering went unexplained, and how powerful collective care can be when knowledge replaces fear. If you carry Rh-negative blood, you are not a mystery waiting to be decoded. You are a person whose biology intersects with history in a visible way. If you do not, you are no less connected to this story. Every transfusion, every prevention, every protocol exists because humanity chose to protect difference rather than erase it. That choice belongs to everyone. As genetics advances, as more people learn more intimate details about their bodies, the temptation to turn biology into identity will only grow stronger. There will be new Rh-negative-like stories, new traits that cluster, new risks that feel personal, new narratives that promise meaning. When those stories appear, remember this one. Remember that understanding is more powerful than explanation. Remember that care is more important than origin. Remember that variation is not a verdict. Rh-negative blood endured not because it was special, but because humanity learned how to make space for it. And that may be the most hopeful conclusion of all. Now, before we end, I want to thank you for staying with this story, from ancient bottlenecks and silent tragedies to modern medicine and quiet prevention. If this narration helped you think, reflect, or simply drift into a calmer space, I invite you to support the channel. Please subscribe if you'd like more long, thoughtful journeys into science, history, and the mysteries that live between them. And if you're still awake enough, leave a comment and tell me where you're listening from, your country, your city, or simply watching from bed. I read the comments, and it's always incredible to see how many places are connected by the same story at the same moment. Wherever you are, thank you for being here. Rest well.